I would like to introduce Dr. Stephen Haley, Vice President and Director at Immudex. And his talk title is Unraveling the Specificity of T Cells and Beyond with Dextramer Technology.
All right, can everyone hear me?
Yes, we can.
Okay, awesome. Thanks very much. And first, just let me say how much I miss our in-person meetings. And I'll look very much forward to seeing everyone on our next really live MetroFlow meeting. But in the meantime, we'll have to suffice with our Zoom here. And thanks for the intro. And today we'll talk a little bit about T cells and moving beyond T cells with Dextramer technology. And so
Stephen, we're still in. We're seeing your presenter view. If you wanted to.
You're okay. Let me see here.
Might be a swap screen function.
How's that?
There you go. Thank you.
All right. And let's do a laser pointer too. So in the next 10 minutes, I'm gonna talk a little bit about why use an MHC multimer in these complex flow cytometry panels, why Dextramer, our reagents, are superior to traditional MHC multimer reagents. And lastly, just a couple slides on integration of Dextramer specificity, phenotype, gene expression, and TCR sequence to investigate T cell responses and perhaps moving beyond T cells in unprecedented details. So first, let's think about the T cell response to peptide antigen, right? And when that peptide antigen is presented to a naive T cell, that T cell is going to blast, it's going to proliferate, and it's going to go on to perform its various effector functions, such as cytotoxicity and cytokine release. But the clonal hypothesis of immunity tells us that each of these T cells, no matter what the function or lack of function, these could be anergic T cells, correct? Maybe all of those T cells are going to express the same T cell receptor. So if we want to measure all of the T cells that are specific for any particular epitope, irregardless of what the function might be, the idea is to go back to this original interaction between the antigen presenting cell, which presents this specificity, this confers the specificity of your immune response, of your T cell response here, this peptide epitope in the context of this MHC to this T cell receptor. So, but if you just chopped out one of these from that very complex aggregation of molecules, that's the immune synapse, and in vitro and tried to stain a T cell with this, you only have a half-life of about 10 seconds. So in order to get around this, MHC multimers were invented. And the first MHC multimer in the class was the conventional tetramer, which was first published in 1996 in a famous science paper called The Phenotypic Analysis of Antigen-Specific T Cells, publication by Jen Altman and Mark Davis. And this was 4 of those MHC peptide pairs that are held together by a streptavidin fluorochrome. And soon after that was the dimer from Schneck's lab at Hopkins there, which is an antibody linkage. A pentamer from a small company in the UK, which is 5 MHC peptide pairs held together by a coil structure of protein. And lastly, there's our Dextramer, and this is our reagent. And the Dextramer is the same MHC peptide pairs as all these other classical multimer reagents, but they are aggregated as put together by a string of dextran, a simple sugar. And this gives us several advantages. So first, the flexible dextran backbone is a simple sugar. So we speculate that this gives us a little more stability to our MHC multimer reagents. And perhaps most importantly, we have optimized a number of MHC peptide complexes here. So we've empirically determined the number of MHC peptide pairs that's going to give us the strongest binding here to our antigen-specific T cells. And these are rare events, so you want them labeled very brightly and very reliably. And lastly, the fluorophores here are covalently attached to this dextran backbone. And depending on whether you get FITC, PE, or APC, which are the 3 dyes that we currently offer, you get a different number of those fluorochromes, which is another method we use to optimize the signal-to-noise ratio. So again, just an optimized number of MHC peptide pairs is going to give us a longer interaction with the target T cells And just to be clear, this is available for both class 1 and class 2 MHC off-the-shelf reagents to find CD8 and CD4 T cells, respectively. So let's look at this old paper here, 2014 now. So this paper is getting long in the tooth, but I like showing this as an example because this was a completely academic publication. There was not a single Immudex employee involved in this paper. And it did a direct comparison between classical tetramer technology versus our dextramers. And you can see both the negative controls are clean. And the high affinity T cell receptors, you can see that both the tetramer and the dextramer will give you a well-separated population. This is a flow cytometry meeting, so everyone knows it'd be very easy to isolate and sort either one of these populations. But when you get into low affinity epitopes, those that are seen in anti-tumor TCRs, tumor-associated antigen-specific T cells, or autoimmune disease, that's when the dextramer will find T cells that disappear into the shoulder here in an MHC tetramer assay. So this is our sort of driving advantage over the traditional tetramer technology, along with barcodes, which we're going to talk about soon here. All right, so here, this meeting is a perfect example. We just saw the OMIP 69 publication listed, and we can see now that 40 colors, maybe even 50 in the future of flow cytometry is not, I should say it's still unusual, but not unheard of in our space, right? So that gives us 40 parameters that you can look at to isolate your cells, to characterize your cells by general flow cytometry. But to look at the overall immune response is going to really require a lot more than 40 parameters. And so we and a whole lot of others now have moved on to DNA barcodes as an additional tag, so to speak, additional label, in addition to fluorochromes, right? And notice here that this is our standard dextramer. There's nothing changed. We have a PE fluorophore, a dextran backbone in the same MHC peptide pairs. But because the size of the dextran, it's enabled us to add a DNA barcode without disturbing the structure of our molecule. And it turns out that this is very important because antigen-specific T cells, whether you're looking at CD4s or CD8s, are pretty rare events. And so to use these reagents and analyze those T cells efficiently within single cell technologies, an enrichment step, and this is good news for all those flow cytometry core managers out there, an enrichment step by cell sorting is still required. We've actually tried this using magnetic isolation instead of the cell sorting step here, but those results have been inconsistent so far. So at this point in time, we still recommend that everyone involved in these single cell experiments to do a cell sorting step. So our general workflow is just to mix your decode, that's our trade name for the barcoded dextramers, decode library together, label your sample, stain them, perform that cell sorting step, and then you can go on into your single cell platforms. And we have our own barcodes, which we call HIT. And we also manufacture barcodes that have been developed in conjunction with both 10x Genomics for their Chromium single cell analysis systems and with the BD Rio dextramers that we have that work with the BD Rhapsody system. Right. And just a few results I want to show from this paper, which is still, this is not completely parsed data yet. This is in the bio archives at this point in time. But and SARS-CoV-2 is still big and affecting all of us. So this paper was interesting in that they used 38 different decode dextramers, and they started with our HIT panel. So these are the barcodes that are specific and manufactured by Immudex. 38 different specificities, plus there were 4 positive controls and 4 negative controls. Always important to include proper controls on these experiments. Your bioinformaticians will thank you at the end. And this, they went through 3 different HLA types, AO2, which is the most common in North America and in Europe, A3 and AO1. And they were able to identify a number of epitopes here ranked in order of frequency in healthy, active, mild, active, severe, recovered mild, and recovered severe diseases. Right. And 15 epitopes or ones they selected as being the most enriched in patients that were affected at SARS-CoV-2. So they elected to take these, and they're going to use the 10x genomics compatible barcodes and look for single cell analysis here. And so they turned out there were 4 really important epitopes, and you can see that some of these epitopes were enriched in severe disease. And the T cells in the most severe cases, they revealed hyperactivation and T cell exhaustion and lack of long-term memory features. But in patients that had mild disease, it was the opposite. And they found T cells reacquired IL-7 and memory cells developed in patients that had mild disease. And not interesting is these 2 epitopes here, the A1 to A1s were both from the spike protein, but these A2 restricted epitopes were both from the N protein. So all the people who've been restricting their studies to the spike protein, because that's where most of the vaccine targets are, may be missing out on some very important epitopes for the T cell response. And we've all seen at this point how the T cells are very important for our resistance to SARS-CoV-2. And so just 2 quotes from this paper, where, as I just stated in summary, the data suggests that the CD8 T cell response to SARS-CoV-2 derived epitopes preferentially leads to short-lived terminal effector differentiation and hyperactive phenotype in severe COVID-19. Well, epitope binding CD8 effector T cells in mild COVID-19 preserve the ability to develop into memory cells after clearance of viral infection. And secondly, the feature barcode technology combined with the class 1 dextamer reagents carrying specific SARS-CoV-2 epitopes and TCR-seq allowed us to study the epitope-specific CD8 T cell response in COVID-19 at unprecedented resolution. And really, it's the resolution that I want to stress here, because as you combine each step of the process and you look at gene expression of the T cells and the high resolution phenotyping of the T cells, and then sequence the TCRs, the resolution of the T cells that you detect goes up each step of the way. Right. And so lastly, I just want to say that now we can take that same barcode, and we've made it available for attaching anything, any protein or anything that you can biotinylate to that backbone. So now people are doing projects like attaching ligands to the DNA backbone, or the DNA barcoded backbone, do proteins for vaccine research. We have projects going in vaccination looking for antigen-specific B cells. And really, you're limited only by your imagination as to what you might attach to a DNA barcoded dextran backbone, so that you can investigate at a very high resolution cell specific for whatever that molecule is that you might attach to that backbone. So lastly, we are hiring. Immudex is recruiting a representative for the Northeast. I was not originally scheduled to give this presentation. But so anyone that might be interested or know anyone that's interested, please contact me for details. And lastly, if people can be intimidated to get these large single cell projects started, our field application scientist, Melissa Thone, is available here in the States to help anyone get started. And she's proficient with the process all the way from FICOL to data analysis. And Melissa is available to help anyone get started or troubleshoot any of these processes on either the 10x Genomics platform, our HIT barcodes, or with the BD Rhapsody platform. And I got to shout out to our scientists. This is an official photo from the back, Kevin Jacobson, who's our senior scientist in Copenhagen that really makes all these processes work. And with that, I'll be happy to take any questions.
Great. Thank you very much, Steve.